So, hello everyone, I hope you had a good lunch. And welcome to this next session called Multi-omics Data Innovation. And let me just grab a few words here. So it is my pleasure to introduce to you Professor Özgün Gökce, Professor at the Rheinische Friedrich Wilhelms Universität Bonn, and a group leader at the German Center for Neurodegenerative Diseases. Professor Gökce is dedicated to advancing brain health and combating neurodegeneration through cutting-edge technologies in experimental and computational neuroscience. No, this is completely wrong. I was supposed to introduce Jeroen de Ridder. Apologies. I had the wrong one. All right, save it for later. So now everybody's awake. That's good. Everyone has a laugh. Thank you. So the first speaker of this session, not the next one, the first will be Jeroen de Ridder, a principal investigator and associate professor at the Center for Molecular Medicine at University Medical Center and a senior PI at the OnCode Institute. He has a very firm background in bioinformatics and machine learning and a strong desire to improve molecular cancer diagnostics using these genomic technologies. And he recently proposed a deep learning method to enable brain tumor classification while the surgery of the patient is ongoing. So I'm sure we'd all love to hear more about that. So please welcome to the stage, Jeroen de Ridder.
Great. Yeah, this wasn't the first time I got introduced wrongly, because there's another Jeroen de Ridder who's a professor in philosophy. So there was, yeah. No. All right, anyway, so indeed, I'm the bioinformatician, and indeed, this is, I want to talk about CNS tumors. So those are tumors of the brain and the spinal cord, and it's a very lethal type of cancer, as you probably all know, in particular in kids. It's also quite prevalent in kids. And in kids, the problem is in particularly big because there's many different subtypes of CNS tumors, and in kids they really all occur. And these are a result of differences in the cell of origin of the tumor, differences in the mutational pathways, and differences in the tumor grade. Now, the first line treatment of CNS tumors is still surgery, where the surgeon takes away as much of the tumor as possible. And in this surgery, this is the first time that you have access to the tumor, so the surgeon needs to make a very, very difficult decision, and that is how aggressive, how much resection should we do? Are we going to do very aggressive surgery, maximal resection, potentially risking very serious side effects, very serious, severe loss on quality of life? But there's also benign tumors where a conservative resection is totally fine. But if the surgeon now makes the wrong decision, you operate too aggressively for a benign tumor, clearly you risk very serious side effects, but the flip side is also that you need to see the tumor twice for a next surgery because the tumor regrows, and so you also don't want that. So it's a very tough decision that the surgeon needs to make. Now there's a lot of knowledge on which tumor types require which type of surgery. So for instance, a medulloblastoma, it's been shown that a conservative resection is perfectly fine and has no difference compared to a maximal resection. But for an ependymoma, for instance, it's been shown that you really need to go all the way and you need to do a maximal resection, because otherwise you're pretty much guaranteed that the tumor will recur and you need a second surgery. And this is why currently there is intraoperative classification of the tumor type for these CNS tumors based on histology. So there's a pathologist during the surgery looking under the microscope to assess what is the tumor type. But this is often inconclusive, sometimes even wrong, and you cannot get a classification at this level of the 82 different tumor types that exist. So what you can do is, but this is post-operatively, you can do a molecular diagnosis, in particular using methylation arrays, but this molecular diagnosis, it takes at least a week, sometimes even 2 weeks. But it's been shown in this seminal paper by David Kapper and his team that using methylation microarrays, the Infinium array or the EPIC array, you can actually very, very accurately distinguish more than 80 different tumor types. And as I said, this takes a long time. So unfortunately, when you get a very accurate diagnosis, the only thing that you can conclude is that during the surgery, the pathologist was right or was wrong. And that's not what you want. So I hope that you see where this is going, is that we wanted to see if we could bring this molecular classification into the surgical theater. And for this purpose, we use nanopore sequencing. In addition to producing long reads, which is what we don't really care about, it's also a very fast sequencing approach that allows you to sequence native DNA, so it includes DNA methylation. And you can get from sample or from a tissue sample to DNA sample for sequencing within 15 minutes. And the beautiful thing is that while the sequencing device is still running, you get already access to the data. So you can already start analyzing your data and potentially already do classifications on the fly. And that is exactly what we try to achieve here. Now, the major challenge, if you want to do this, is data sparsity. The nice thing about a microarray is that you get pretty much 850,000 CpG sites in one go. Now, with the nanopore device, you get a very sparse picture. So you cannot sequence for too long. I mean, you clearly can get full genome coverage, but this will take maybe 24, 48 hours or so. And we only have, well, 30 minutes, maybe an hour or so of sequencing time. That's the window of opportunity that we have. And as you see here illustrated, you get very, very sparse coverage. I always compare it to, if I show you a picture of two parrots, right? But now I take away 99% of the pixels. So I only give you 1% of the pixels, and I don't tell you which pixels I will give you. Then you still need to recognize that there's two parrots in this image. Yeah, now, as a human, you probably fail this task, but computers actually can be trained very well to deal with this missing at random data. And that is what we did, is we trained a deep learning classifier that contains a couple of fully connected layers. But in these models, there's a lot of parameters to tune, about 150 million parameters in this model. And the reference data that we have at our disposal, the currently available, publicly available data set, that's the Kapper et al. Data set, which contains about 2,000 reference microarray samples across these 80-something classes. So there's no nanopore data available at all, actually, only microarray data, at least not in any reasonable quantities. And then we had this issue that we needed to deal with the extreme sparsity of the data. So we need to be able to train models that are robust enough to deal with this amount of sparsity. And this is what Carlo and Mark developed. They said, okay, well, maybe we can simulate training data and in this way generate much more of it. And also make the translation between microarray data and the nanopore data. So what we did is we took this Kapper et al. Data set and then we simulated nanopore reads in the genome, and we looked into microarray data. Okay, what is now the methylation state and this locus, right? We add a bit of the errors, we add all the nanopore read statistics, read length, error profiles, these kinds of things. And in this way, you can generate a realistic data set that contains a sample as if it were sequenced on a nanopore using, let's say, 30 minutes or 40 minutes or 50 minutes of sequencing. Yeah? And you can generate vast amounts of relatively realistic training data in this way. And that's actually what we did. We generated about 45 million nanopore runs, and that is the amount of data that you really need to effectively train these very deep neural networks and to teach these neural networks to cope with the extreme amount of data sparsity. Yeah, so this was trained for a couple of days on a quite sizable GPU, a set of GPUs. Of course, we split training data, test data, and then we can validate. And we can also do these simulations to generate realistic validation data, right? So for cross-validation purposes, and see if compared to the EPIC array data, which contains all the CpG methylated sites, if we can still make a classification if we have very little data at our disposal. This is the results for the simulations. You see this is a confusion matrix, and you see a very strong diagonal indicating that for the vast majority of the cases we actually do a very good job. The F1 score is above 90% for the top one, and for the top three classifications it goes to the 99%. Now, this is of course all simulated data, and then the real question is, can we now do this on real data? Now, does this work on real nanopore data? Now, this project was in full collaboration with the Princess Maxima Center for Pediatric Oncology, and they had the vision to really create a biobank. Yeah? So they have a ton of samples, for historic samples, biobanked in their freezer. So we took pretty much all of these samples one by one and nanopore sequenced them and accrued data up to 1 hour. And this is the result. So here on the left you see one of the classes clearly is directly the winner in about 10-15 minutes. It gets to the right classification. Here's the second one on the top right. It also in 10 minutes or so goes to the right classification. On the bottom right you see that in the beginning there's a bit of confusion. Yeah, it doubts between a few, but after 30 minutes of sequencing, also one of the classes is the clear winner, and in all of these cases it turned out to be the correct prediction. Now, we repeated this for pretty much the whole cohort, so 50 retrospective cases, and out of those 50 cases, 46 reached the correct diagnosis in 40 minutes of sequencing. The average was about 20 minutes of sequencing that's required. And there's 4 unclear diagnoses. Those are cases where we do not reach the 95% threshold that we typically require. But that's always better than making confidently the wrong diagnosis, right? So we never saw that in the 50 retrospective cases, luckily. Now the beauty is that this is pretty much all you need to achieve this. Yeah? Very small setup that you can put anywhere next to the OR, if you will. We actually put this next to the OR in the Amsterdam Medical Center because we also included some adult samples along the way. But okay, this was all retrospective, controlled environment. And so we said, okay, let's also see if we can do this for real, right? So we asked, okay, when is your next surgery at the PMC? So can we not do a complete shadow test for real, where we do everything in real time? And we filmed the whole procedure. Yeah, imagine Carlo working under all this stress with 2 cameras in his face. But yeah, we have the footage now, so I'll show you the shortened version. So this is Ari, a head technician in the PMC. He receives a call from the OR. This is in the PMC. The OR is actually at the UMCU, which is a little bit of a walk, so we can save some time there. So we collect the sample, it's brought back, and then we do a bit of sample registration, so a little bit of administration, so the sample gets a proper barcode and an entry in the system. And then Ari cuts a few pieces. One goes to the pathologist for the histology, right? So that's as it is normally going. But then one piece goes to us. Yeah. So this is Carlo. In the background, it's Mark. And that's me. Yeah, and then we can start with the DNA isolation about 20 minutes after we got the call from the surgical theater. And this is a process that takes about 20 minutes. We do some quality controls. And then we can start with the library prep. We use the rapid prep for this purpose, which takes about 10 minutes. And after 10 minutes, we can start with the sequencing. So 50 minutes after the call from the surgical theater, we can actually start with the sequencing. And then 10 minutes later, we get the first data and we can make the first classification, which after 10 minutes of sequencing is not yet accurate, but then the confidence starts growing. And in this case, 80 minutes, the whole procedure took about 80 minutes. 30 minutes of sequencing was sufficient to call very confidently a schwannoma, which later we also checked, of course, with the EPIC array data. Turns out to be the correct class. Now, this was, of course, very nice. It all worked, also in real life. And this means that the surgeons, they get very confident about it. Even NS1, they say, yeah, let's do this more often. So they requested this more and more, and in the beginning especially we did this in complete shadow testing, so we did not inform the surgeon. Now I have to say that we do actually inform the surgeon, because the surgeons think it's unethical to withhold this diagnosis from the patient. But in the meantime, now we've done more than 25 intraoperative cases. These were actually not reported back to the surgeons yet. And this is all done in practice, real time, as if it was reported back. And in 72% of the tumors, we are able to correctly classify the tumor with at most 45 minutes of sequencing. And the nice thing is that we do not see any misdiagnosis yet. There's a couple of cases where it's unclear, so that the classifier is not confident enough to make a diagnosis. Most of the cases, this is due to low tumor purity samples or to classes that are just not in the classifier. There's these very rare exotic cases where also the pathologist doesn't know what it is, and clearly also the classifier doesn't confidently call it another class. So that's good news, I think. All right, so this is now all published, so you can actually do it yourself. All the code is available at this GitHub link, and we've actually set it up now in collaboration with a couple of labs worldwide. We're talking to a hospital in Israel that's been using it, in Austria, in the US. So it got a lot of attention as well in the media. That also helped. And so the nice thing is that you really quickly from the original idea, you can bring this now into the clinic, and it's actually used in academic centers in a few places. Now, if I have 2 more minutes, I just want to talk one case. I mean, I'm not a clinician or something, but I still think this is a very telling case. It was a boy with an intramedullary tumor. And so there was an MRI beforehand that suggested ependymoma or an astrocytoma. So that means that's a tumor originating from the central nervous system, so central nerve cells. So the intraoperative histology was consistent with that, so they did frozen section histology, and you saw this clustering of cells, which is also suggestive of ependymoma. And then we ran our classifier, which we call Sturgeon, and it said with a very high confidence. It was a bit difficult in the beginning, in the first few minutes of sequencing, but after a while it was quite confidently called as a schwannoma, which is a tumor originating from the peripheral nerve system. And then, the pathologist kind of laughed us out of the room, and he said, well, AI is not everything. But for us, this was a beautiful example to learn from. And so we did pretty much everything that you can think of post-operatively. So we did FFP histology, and there it actually said, it could actually be a schwannoma. We did immunohistochemistry, everything fits with schwannoma. We did the EPIC classifier, and it showed the VGL fusion, which is typical for the schwannoma. We did the RNA expression analysis that showed this fusion actually existed, and we did the RNA expression classifier, which predicted it to be a schwannoma. So in the end of the day, also the pathologist concluded that this was a very rare case of an intramedullary schwannoma, which only occurs in 0.3% of the cases. And this is to show that these classifiers, they don't suffer from these biases for the fact that this is a very rare tumor type, and it just goes with what the data tells it. So with that, I would like to close this talk. These are the people that did all the work from my lab. Mark and Carlo did all the heavy lifting pretty much, but this was in full collaboration with the PMC, Bastiaan, Leonard and Peter in particular. But also the Amsterdam UMC played an important role because they, in response to some reviewer questions, also showed and provided the adult patient samples to allow us to show that this also works in the adult setting. Thank you.
Thank you very much, Jeroen. Maybe there's time for one short burning question.
Yeah, thanks, Jeroen. Wonderful story. This is a multi-omics conference. So how would you compare this methodology with things like the iKnife, where you can do the mass spec analysis of of materials which are vacuumed out of during the operation?
Yeah, so I think this is, can go side by side. I think that, so the iKnife actually, real time gives an indication of where the surgeon is cutting. It doesn't, I think, tell you necessarily at this level of detail what the tumor type is. I think it can certainly go side by side. There's also a study that will come out in Nature pretty soon that showed that you can do intraoperative imaging, for instance, of a small tumor section to also give an indication of if there's still tumor infiltrating cells in your section. So I think all these modalities ultimately need to come together to get to one optimal diagnosis that optimally supports the surgeon in the decision making, because clearly these are very, very important decisions, especially for the brain. You can have access to the tumor. You want access to the tumor only once, right? And you want to get it right.
I'm afraid to have to cut it short, but I'm sure Jeroen will be available for your questions in the next break. So thank you very much. Then our next speaker. So our next speaker, Professor Özgün Gökce.
Oz, everybody calls me.
Özgün, sorry. Yes, as I mentioned before, professor of the Rheinische Friedrich-Wilhelms-Universität Bonn for neurodegenerative diseases, combating neurodegeneration, studying brain diseases, and he founded this research group at the Ludwig-Maximilians University of Munich, and prior to that he worked as a postdoctoral researcher at Stanford University. So please join me in welcoming Professor Özgün Gökce.
Thank you very much. Thanks for the invitation. And I'm really lucky to start with after such a nice talk, and we are talking about using the integration as well. But first I wanted to thank the people actually do the work, my group, and one key collaborator is Martina. Martina, she actually ran the electron microscopy where we combine with the spatial transcriptomics. About my group, we are focused on aging, and specifically brain aging. If you look aging, all the incidence of the bad things increase, but dementia shows a different slope, and there's an inflection point. And my research, when I started my group 2016, was decided to focus on that inflection point and what is happening there. And differently from the others, we use Richard Feynman's one of Feynman's talks. There's a plenty of room at the bottom, where he suggested biologists should use more mathematics. And thankfully, we had developed single-cell transcriptomics methods, and my focus had been white versus gray matter regions of the brain. And this work had been, first we identified with single-cell white matter-associated microglia that actually consort program to digesting damaged myelin. And to our surprise, actually, a follow-up study showed that this microglia increased T-cell infiltration and induced brain aging-induced brain damage. And on our last work, we also showed in the neurodegeneration, this happens way earlier in the Alzheimer's disease models, and we could actually show that this, a little bit more moved, we should show that this induced, and there is some movement in here, we show that the T cells in the earlier 5XFAD model induce a DAM4 type of a microglia state, and using the spatial omics here, you see in the merfish, we show that these cells are, sorry about the slide, kind of moved, but these cells are very close to the T cells. And if we deplete the T cells with a T cell antibody, we can actually decrease this small population on the tail of the UMAP in here. And this slide is destroyed, so we move to the next one. What I wanted to show that using the single-cell transcriptomics and spatial omics, we make a very quick jumps from the region to the molecular mechanisms and the cell types. And Richard Feynman again felt that in his talk, if you wanted to make even more progress, just make the electron microscopy better. And Peter in my group thought how we can do the electron microscopy better. So he thought, we don't have the labels electron in electron microscopy. Can we carry the labels from a spatial transcriptomic method? And we can't, because EM is not forgiving. Protocol is very tight. So we did two adjacent sections, and using this proximity, we hope to transfer the knowledge to each other. We kind of build a bridge between the adjacent sections. So we induce a lesion in the white matter by the detergent injection into the mouse brain, and took one section to the MERScope and one section to the electron microscopy. MERScope is a really powerful methodology, and we use the MERFish in a MERScope machinery. And here you do a serial in situ imaging, and with the 16 cycles, with the multiple barcoding, we are 100% confident we can label up to the 10,000 probe transcripts on a section. But for that, you need to do some tissue enlargement, because the crowding became an issue. And what you get here is with only 300 RNAs, we can already reconstruct and each cell in a tissue and see which cell states are there. Of course, in order to understand the sensitivity, we did in the same cohort a single cell transcriptomics specific on the microglia, and you can see here MERFish and the single cell RNA sequencing for the cluster markers quite agrees. But this is kind of already developed and shown. So the Martina took the adjacent section, embedded in the raisin, and we cut one of the largest EM screens, 7 to 4 millimeter regia, 200 nanometer thin sections, 20 of them cut from this section, and scanned in the electron microscopy. Here you see the MERScope cell annotations on the adjacent section overlaid to the electron microscopy. And you can see the electron microscopy quality is quite good. So with the MERFish, we can actually identify cells unbiasedly. In the EM, Martina will go back and quantify which are the cell states in the lesion area. What she showed is here with the MERFish, automatically T cells around them, interferon response microglia clusters. So we again see this interferon response and T cell linkage in this model too. But this was qualitative. To switch to the quantitative, we did something, actually Hanyi did something heroic. She went through the EM images, and on these 20 sections, each microglia, she segmented for these 8 features. And from these features, we could get a ultrastructural identification of the cell states. And here you can see this UMAP. This is not a transcriptome. This is all ultrastructure. And you can see each immune cell in the lesion area, how they distributed. And you can learn about what the cell is doing, because transcriptome is correlative. Here we see the organelles and what the cell exactly doing. We have a functional window to the cell states. Now we have a single cell. Single cell to MERFish integrating is quite easy because we have the same anchors. We are measuring the RNA in both cases, so we integrated these two. We didn't know how to do the EM to MERFish integration. We did what we could. We just take the maximum projection of the two sections and average the neighbors of EM to the MERScope transcriptome to the structure. And that averaging, did it work? So we asked the question, what are the genes correlate with endoplasmic reticulum? Endoplasmic reticulum genes, the structural genes, are the highest correlating genes. For the mitochondria area, mitochondrial matrix, heterochromatin SYN3 and heterochromatin are the first two figures coming. So there is some type of a logic. And this is one of my best slides ever. So in the column, you see the ultrastructure, and on the gene panel top, correlation with the genes. It's not a foundation model, but a Spearman correlation, just showing us there is a tight correlation and much more diversity when we combine ultrastructure and transcriptome. And both of the integrated data set, imputed transcriptome or imputed structure, correlates with that, and they hierarchically cluster according to the cell states. So there's a plant of room at the bottom is the Feynman stalk, and we can cut 200 nanometer sections, so which one cell can give us a 250 sections. So there is a plant of room down there. And one of the reasons, I guess, Martin, we working with the Martin, I love lipidomics. Brain is highest fat tissue after the lipid tissue in our body, but we don't understand that lipids. And with Martin, we are trying to correlate what we know from transcriptome project into the lipidome. And with the Jianshu, we thought, yeah, correlation is great, but can we develop a generative model? And what it's here, we are still working on that, but using the transcriptome, we can actually predict, and quite accurate actually for the expert eye, electron images, and if we cut the cells, we can actually predict the cell states from the EM2. So this is already ongoing. And I wanted to say, actual Feynman is, I believe, to looking to the thing from multiple angles is the right way to accelerate the process. And for me, the main point is within a very limited time from white matter to clinically ready drugs came out to be from this kind of multi-omic data integration approaches. So I finish my talk here. Am I on time? Yeah, time, almost there.
Right. Thank you very much, Özgün. I'm afraid we have to cut the questions in this case, but feel free to talk to him at any time. Here's your chocolate. Thank you very much.
Okay. The next person I would like to call to the stage is Horvatovich. Well, I don't think he does need a lot of introduction in this audience, so he's very active in the cross-omics data analysis, integration and stewardship pillar. As a professor of computational mass spec in University of Groningen. And well, he doesn't do only computational work, but has actually also running the mass spec lab in Groningen. And well, he combines the two and will talk to us about proteogenomics. So the floor is yours.
Thank you very much for the nice introduction. Indeed, one of my biggest interest is basically to understand how genomic variability affects protein, not only expression, but also protein sequence variants, which is called proteogenomics. And this work is, I'm not doing alone, I'm doing it with Viktor Guriev, also sitting in the audience. And he's representing the knowledge of the genomics and transcriptomics world. I am the mass spec and proteomics, and we work together over the years to develop a proteogenomics pipeline, which is trying to predict protein sequence variant from transcriptomics or genomics data, which can be used to accurately predict which type of protein sequence variants can be detected when you collect genomics or transcriptomics and proteomics from the same sample. So just as a generic introduction, right, so I think in this audience is quite obvious the information flow in a living system flowing from the genome right through the transcriptome, which is providing a proteome, which is having some fulfilling some biological function, which can be enzymatic, receptor, or structural one, and basically together also with the environmental factors like stress or pollution, but also lifestyle, can determine the disease and the phenotype. And the question is how, right, the genomic variability here is affecting proteins and protein function. Of course, this is not the only omics layer. You have many other type of omics layer. So if you think multi-omics, now it's growing families of molecular layers. If you look at genomic variability, right, you have population-based genomic variability between individuals. We all are a little bit different from each other, which represented by population genetic variability. If you take, for example, a cancer patient, especially melanoma, which is very heavily mutation-driven cancer, you can have different clones which have different mutation, so you can have also within a patient variability of your genome. And of course, when you have this type of genome variability, some of them is affecting the protein coding function, and some others is affecting just the regulation of the protein expression. So when you go to the genome reference, right, you can classify all mutation from single nucleic acid variants as the most easiest one, to insertion deletion, to multi nucleotide variants, and also other type of complex genomic rearrangement. And of course, you also have some portion, even in humans, so-called non-reference segments, which can still lead to protein products. And most of the often lead or forgotten from even transcriptomic processing pipeline, but also from proteomics, if those are expressed and if those have a function. One of the biggest variability is splicing, and splicing can be affected by many regulatory factors, and basically it's affecting of how many protein variants do you see from one protein coding gene. And of course, people doing proteoform analysis or long-read sequencing can have access to this type of information. All right, so if you go from genome to the proteome, there's also some other type of rearrangement, and here I'm showing you this in order also to explain why we are choosing certain type of transcriptomics data for proteogenomics analysis. So if you go from the genome, right, you get a premature mRNA with introns and exons, introns and exkeys, you have polyadenylate end capping, splicing, you have also RNA editing, which can also modify the original sequence, and you have the translated proteome. So if you want to do proteogenomics from one exon sequencing or whole genome sequencing, the problem, you don't have the splicing information. So you need to predict that with tools, and it still remains a prediction. Other choice that we have, right, is next-gen sequencing with per end or single end, which is still bottom-up type of sequencing, or having long-read sequencing. We are still using the next-gen sequencing, so the bottom-up approach. This means that if you have mutations which are very far away apart, right, we cannot, we only have the combination of them, but we don't know which exact combination is in the proteome. So polyadenylate transcript, they are matured ones, so they are the most probable translated transcript, and this is the reason why we have chosen to develop a pipeline for polyadenylate transcript. And of course, it's possible still to integrate with that the ribosome sequencing, which contain the sequence that are on the ribosome cut, but only with 30 base pairs, which are the ongoing translated protein. And you can also have a stable isotope labeled amino acid to see the new translation, which can also be possible to combine with our data sets. So why is problematic in proteomics to detect protein variants? First of all, because so mass spec-based proteomics is not a sequencing. It's just you take a protein, you digest it to peptide, and you just smash it. You use a fragmentation method, which is base rule, but still stochastic. So sometimes you don't have any fragments between two amino acids, so you cannot so accurately determine what is the exact protein sequence. And for that, the normal proteomics workflow use high quality clinical sequences with 20 sequence or some reviewed or non-reviewed
isoform sequence. So in total, you have around 20,000, which is available in public database. But we still miss many of those. So then also the problems is that, for example, if you do data-dependent acquisition, if you look at the isotope clusters, which is non-fragmented, and you do some fragmentation with DDA, even DIA, LCMS, you only identified a fraction of them, which is most of the time is 30–40% of the data. So one part can be due to the missing sequence from this canonical sequence, and the other part is post-translation modification. So what you can do for identification of the protein sequence, you can do proteogenomics that we do with DNA mRNA prediction. We can also now use deep learning method to predict, to use de novo sequence and predict fragment spectra, and use mutation database, which is not complete as well. For PTM research, you can have restricted research, which is most of the case done, or open modification search, which is looking for any type of modification in the protein. And of course, this is providing a large complexity, and often sequence variants and PTMs are confounded in this space. So the approach that we, so I will show you also to study on the proteogenomics approach. So the pipeline that we have developed with PhD student Yannick Heihemeyer is basically use polyidentity transcript, but also which can be adopted to other type of genomics information, which take, after quality control, the transcript information has a sequence alignment to the non-annotated genome, has a de novo transcript assembly, has a tool for alternative splicing, and annotation also of the known mutation using web and GitCat tools. After that, we make a SAM-specific protein FASTA database, which is also annotated with all the information that we get from if the mutation is already known, and which is used in our proteomics pipeline. We are using FrapPy for that. You can have database search there, but as well DIA processing tools. And we have some type of quality control methods. So the type of annotation that we get, right? We have different type of sequences. First, we have a few which is annotated with the variance effect predictor in frame or frame shift. You can see also here which type of annotation we already get, which is very useful information to interpret the type of mutation, but also the localization and which database there is more information on it. And for some of those mutations, we have also allele frequencies, so which is showing in large population studies the mutation. For the unknowns, we can have unannotated transcript. We can still have some homology search that we are also performing. Or if you have a new ORF from de novo transcript assembly, we can also have some type of guess with homology search, which type of sequence we have. All right, so I want to show you two studies. One that it's a little bit older study, but show all the steps of proteogenomics and also the advantage of it. So it's related to COPD, which is a respiratory disease. And it was a study done with stage 4 COPD, which is end stage of COPD, when people are hospitalized and basically need lung transplantation. And it's included controls. Those are people treated with lung cancer, but it's allowed to take a little bit of biopsy apart, which can be used as a control. And then we did deep proteomics and also deep next-gen sequencing of the polyidentity transcript. So one of the highlights that I want to show is that many people are using still in the clinics mRNA-seq as a proxy for protein levels, which is there's a medium correlation, but not every protein is showing the same type of elevation or level than the transcript. And you can see here, right, in each type, which fold change you have between the two groups. And many times it's going in the same direction, but you have some times where it is different. And I want to highlight only one protein here in this protein-protein interaction, network, which is in a central hub, that's SUMO2, which is a stress factor. It's also labeling the proteins like ubiquitination, but it has a very important function for nucleus transport, transcriptomics regulation, and so on. And based on transcript level, this is not differential, but it was highly downregulated in COPD patient, which is showing that it will be not found with the transcriptomics. So what do we gain with our proteogenomics pipeline? And if you look at the number of peptides, most of them are mapping to public databases. So the new forms or the new protein variants is very few compared to the overall number of peptides. But this can be still very informative. So if you look at which of those variants are only was found in one of the groups, so controls or CPD, with certain number of matched mass spectra, and here you see also the number of patients in which it was identified. You see very interesting genes and proteins. Many of them in the control was related to immunology, and I just want to highlight one or two from those sequences. One is SORPS2, which is completely a new exon identified in SORPS2, one gene, and ACAP12, which is a single amino acid variants found in two peptides, which was already well known to be related to CPD. Here you see again those peptides, and here highlighted most of them are single amino acid variants. Here you have also the protein identification scores with the two peptides. And basically, this two peptide is mapping to a new exon between exon 8 and 7. And here you see the sequence which was obtained from the transcript with the two peptide matching to it. And of course, when you have proteomics identification and you have a new finding according to Human Protein Project, you need also to validate that with synthetic peptide, which we have done here. You can see that the two mass spectra is corresponding very well. Basically, this new exon is providing a new protein domain, which is providing toxicity for this protein. So what is the exact function in CPD? We don't know yet, but this is providing already a good hint for further study. The other study which I want to speak about is still ongoing investigation, so I only have partial results. But it's basically related to cutaneous melanoma when we had 24 different patients post-mortem. So the pathologist, after the death, went back to the bodies and collected all the metastases of those patients. So here you can see two patients with the different metastatic location. And also we have extremely rich clinical annotation, clinical history of this patient, what was the survival rate, which type of treatment the patient obtained. So with our pipeline, we have analyzed the non-reference peptide, and since it was post-mortem sample, the RNA-seq was quite degraded. We still get some high-quality RNA-seq to be able to perform the proteogenomics analysis, but we are seeing much more of those variants when we do from fresh sample. So one conclusion was that there is a lot of patient-specific mutation. So mutation can be extremely patient-specific, and there is a very few number only shared between patient. But within patient, it's not shown in this graph. Between the metastatic, we see much more similarity between the different non-reference peptides. Yeah. So which are the most mutated genes? You can see it here. And many of them are novel mutations, so not known was or not annotated, found in the public database. And here is the distribution for which type of mutation it has. So many of them is linked to splicing. And we can also, if you take the pathway analysis, many of those pathways are linked to known cancer mutation, and those genes are also highly connected. Finally, what I want to show in this study is basically how those mutations are also linked to different molecular phenotype of melanoma. So what we did from the proteomics profile, we make hierarchical clustering, and we identify 4 clusters which more or less behaving the same. And you can see also that some of the patients have metastasis, which is classified in different of this hierarchical clustering classification. And this marker profile can be used to predict new treatment. And you can see that some of these mutations are highly specific to those profiles. What is the effect? We are now investigating that. And finally, as a future perspective, I just want to mention one clinical case that we had with the Consortium of European Cancer Moonshot. It was one patient that was already in a hospice, and they have predicted only a few weeks of survival, came to the laboratory in Lund, and basically provided sample. And you can see it already that they have identified 6 different regions, which was expected, which also see different cell structure, and most probably those are different clones. And with the image analysis software, with segmentation, and also getting different type of descriptors for the structure of the cells, it was possible to determine that basically it's constituting of different cell types, these different clones. And based on the molecular profile, it was possible to suggest a modification of the treatment, and this extended the life of that patient with 20 months. I wanted to close this presentation also with some highlight that we want to continue with proteogenomics. We only did it for book proteomics, but for spatially driven clonal proteogenomics and use that for personalized medicine. Also including neural networks or marker machine learning-based spectral prediction to improve the accuracy of the peptide variant prediction. Also study the interplay between the marker variants and PTMs, and basically also to study the effect with 3D modeling using AlphaFold and CRISPR-Cas validation. So this work has been done with colleagues at University of Groningen, at UMCG in Rima, and also the members of the melanoma cancer moonshot. So thank you very much.
Thank you very much. Good to see that we are moving a little bit into the direction that Albert Heck wanted us to move into. Time for one quick question from the audience. If not, then I'd like to thank you again, and we have some chocolate for you as well, of course. And then I'd like to call to the stage Sukhvinder Nicklen. She's representing PacBio. She is commercial manager for Europe, Africa and Middle East, but has a special interest for the clinical implementation of PacBio sequencing in clinical diagnostics. And the floor is yours.
Thank you very much. It's great to be here because of the really important relationship we have with Radboud and the collaboration. Nijmegen is almost a second home for me, so I'm always happy to be here. Now, those of you that know us know we're all about the highly accurate long read, but I'm not going to talk about the science behind that today. I'm going to talk a little bit more about the relevance of omics in our world. Oh, which one is it? It's this one. That one. Thank you. So I still find it amazing that in each and every one of our cells, there's a huge amount of DNA packed. And if we removed that and stretched it out end to end, it would be 2 meters or so tall, so way taller than I am, even with my heels, and why I'm not standing behind this. Now, the important piece about this is for it to be able to fit, as many of you already know, it's tightly wrapped with histone proteins and forms the chromatin structure. Now, the method that you see here on this slide is well established from a number of years ago, FiberSeq, and it uses the enzyme methyltransferase, which goes along and methylates all of the A bases that are available on the open chromatin. So this is really important because those are the pieces that will interact when it comes to protein expression. Now, this is the piece for ourselves in terms of omics for the PacBio world. We already can provide a highly accurate long-read genome In addition to that, you get the Metabolome for free. So the data is there and you can easily access that. There's no need for any biological modifications. I've just briefly mentioned the fiber-seq, so you can also get the chromatin epigenome. And then by following our protocols for RNA-seq, the Kinnex protocols, you can actually get the transcriptome as well. The really important piece of this is that you can now do this all on one flow cell in a single sequencing run. So this is described by this paper here by the group Volga et al. So how does this actually work in practice? You have your DNA and your RNA. The DNA undergoes the fiber-seq protocols, and from that you get the 3 omes. And at the same time, you use the RNA with the Kinnex protocol to actually get your transcripts. And all this information comes together from the Revio run. You then can get this type of really, really complex information all layered together. And one of our founder members, Jonas, said to me last week, he's waited about 20 years to see this level of detail. Now, I don't really have time to give this justification right now to give you detail, but essentially what you see here for this particular part of the genome are 2 haplotypes. For each of those haplotypes, you see 4 pieces of information for the omes. And what you can actually see on the first haplotype, haplotype 1, there are 2 transcripts. If you look at the methylation and you look at the chromatone profiling, you can clearly explain why there is that longer haplotype on the first, the longer transcript on the first haplotype, but on the second you only see the shorter one. Now, all of this kind of information is believed to really, really show relevance for the area of rare disease and beyond. So on that, the application that I'm showing you here is within the UDN. There's some color coding here for the genome, the methylome, the chromatin, and the transcriptome. And if you look at the different clinical phenotypes that are listed on the right-hand side, you'll see that one or more of those can be attributed and help to understand what is actually going on within that patient. And I think we're going to see more and more of this as more individuals are working in this area. Now, I think this is a very short taster on what is possible, but one thing I would like to see is we are living in an era where there is transformation. Alex earlier talked about the changes that are happening with long read, and there is no question at all that short read NGS has made huge discoveries. However, in the last sort of 2 or 3 years, where folks were working on our predecessor instrument, the Sequel II, there were really, really obvious that there were some challenges. So there may have been gaps, some errors were made, there were collapsed haplotypes. Now, since the start of the Revio being out in the field, out with people like yourselves, what is clear is that you can actually address these areas and that you no longer have to compromise between the high data quality that you want and the throughput. And what we're seeing with the Revio is that we're really entering another phase of what NGS could look like. I think with this, please do come and speak to all of us if you have any further questions on what's possible, if you have any questions around the science behind the long read, but certainly in terms of omics, I think it's a really exciting time and we're going to see things expand as the next year and so follows. Thank you very much for your time.
Thank you. There is time for a question. Maybe I'd like to give you the chance to elaborate a little bit on the Kinnex. What is different in Kinnex from the regular RNA sequencing, for example?
So I think if you come to our booth, I'll run through that with you yourselves, so if that's okay.
Okay.
Thank you.
Well, then we conclude the session.
Thank
you very much for all your talks.
Oh,
and Alain, you can introduce the next speaker.
thank you.
Yeah, thank you, Peter Bram. So the next session is our keynote. And as I mentioned in the beginning of the day, our keynote is Professor Rima Kaddurah-Daouk. And unfortunately, she couldn't be here physically, although she really wanted to, but of personal circumstances, again, as she had to be there tomorrow in the US. And unfortunately, we cannot time travel and space travel that fast, but I'm really glad that Rima can join us here online. Hi, Rima, can you hear me? I guess You can, right?
Yes, I can. Can you hear me okay?
I can hear you fine, so that's fine.
Okay.
So Rima is a professor at Duke University in the departments of psychiatry, behavioral science and medicine. And she has taken a pivotal role in establishing metabolomics in the biomedical field. Started several initiatives, spin-off companies like Metabolome, founded or co-founded a Metabolomics Society, had several large fundings acquired in that sense, but also started journals in this field, which is incredibly important. Applications are, among others, in Alzheimer's field, and there she moved on to things like the pharmacometabolomics initiative, and slowly got into like contacts with the genome, the gut microbiome, the exposome, and all resulting into big data that can predict basically the health for every individual. Well, that's a lot to cover in half an hour. So Rina, I'm gonna ask you to take that challenge to take us through all these excitement developments within a half hour.
And I thank you very kindly. Just want to make sure you can see my slides and you can hear me okay.
Yes, we can. We can hear you.
You can.
See your slides.
And indeed, my sincere apologies that I was not able to be there in person. I truly tried. It was impossible. I apologize for the audience. So my apologies and my best wishes to all. And thank you very much for your kind invitation. The Netherlands is second home for a lot of the work that we do in the United States. Our bonding is very strong. I hope I can exemplify from very large initiatives that are funded in the United States, where our focus is on the metabolome. The metabolome, the collection of small molecules that constitutes our biochemical pathways, signaling processes that you see on the right, and the important influences of our genome, our exposome, the diet, socioeconomic factors in our lives, the gut microbiome. All of these influence our health, provides us with a unique identity for each individual, and imprints our metabolism, where the metabolome is a readout. It truly is the final readout of all of these influences on our health. So it's very important to try to understand, if we are using an interconnected approach for the net influences of the omics, that we put the metabolome as a readout at the end and ask how did each one of these affect our metabolism and contribute to susceptibility to disease or to the disease itself. Of great importance for us is to look at an N of 1. Each individual, what is the best paradigm to preserve health in each individual? What is the disease trajectory within each individual? How did we arrive at a disease in different ways? So the N of 1 is really the ultimate goal in putting this big omics data with the metabolome as a readout. The things that I will share with you are snapshots today. Unfortunately, time is not enough, but I want to give you a bit of a snapshot. How does big science happens in the United States to connect the omics? My personal experience in seeing how we are building resources to use the metabolome as a readout for the genome, the gut microbiome, the diet, the exposome. Number 2, how are we running big initiatives such as the Alzheimer's, the AMP-AD, Accelerated Medicine Partnership, a big initiative set by President Obama, where we are embedded as multi-omics teams. How does this big science work? How do we make data public? This is a first example of open science in the biggest scope that has happened in the United States. I want to tell you a little bit about the gut microbiome initiatives, precision medicine initiatives, mood disorders, ending with one or few minutes on the systems approach, knocking boundaries across diseases, where we are not thinking now about diseases with silos, within silos, but common mechanisms that affect result in many diseases or contribute to many diseases. I want to exemplify with a large initiative that has been launched for inflammation, immune dysregulation as a common paradigm across diseases, psychiatric, degenerative, and metabolic disorders. So a bit of a flavor, and at the end of the day, it's big data, it's fair data, and the Dutch has truly pioneered the fair data, and many of the participants in this audience taught us about the use and the reuse of the data, the importance of it. So we look back to the Dutch as we build all of this, and we partner with the Dutch. It has been a wonderful journey working with many of you. We want to redefine and reclassify a lot of the diseases that we study. So the consortia that we lead, this is over $85 million invested in these large consortia, the Metabolomics Consortium for Alzheimer's, Mood Disorders Consortium, Alzheimer's Gut Microbiome Initiative, and the pharmacometabolomics that we started a while back that taught us a lot about drugs we take. How do we respond to medications? How can we optimize the use of medications for each individual? So each one of these is a journey and is a large number of scientists, resulting with over 120 scientists embedded in trying to articulate this vision that I will share over the next few slides. So first, let us talk about how do we use the metabolome as a readout for the net influences of the exposome, the diet, the gut microbiome. Important, what are the tools we are building? And we are building collaboratively with the communities. Number one, we need metabolomics tools that can measure and quantify a large number of metabolites, whether they are amino acids, bile acids, lipids, neurotransmitters. So we, as metabolomics community globally, we have been pushing boundaries to try to measure quantitatively, if we can, a large number of these metabolites so we can complete our metabolism and our understanding of these metabolic processes that are disrupted in disease and signaling cascades. And you guys in the Netherlands, of course, have this wonderful center of excellence with Dr. Thomas Sankemeyer that has been scaling and innovating and has been a partner in many of our consortia as one center among others that we incorporate within our consortia to bring 8 or 10 platforms so we can enlarge our horizon of capturing metabolism. And this is on the analytical side. We very much partnered with the Germans at the Helmholtz Institute to define the influences of the genome on the metabolome. If there is a metabolic defect that we see that is linked to a disease, we want to know if there is a genetic base. Does the genome contribute to the disruption in metabolism? And hence, our German partners who built phenomenal atlases connecting the genome and the metabolome are continuously embedded in all of our consortia because they are the ones who are teaching us, enabling us to understand if there's a genetic base for everything that we are looking at. The influences of the gut microbiome is big on the metabolome, right? This bacteria that lives with us, we're going to comment on it across our talk. But these are some of the initial papers, foundational papers that came about that taught us indeed the gut microbiome imprints and tells us something about our metabolome in real time. And we have been on it, trying to understand how bacteria that lives within us can complete our metabolism and how we can complete the metabolism of bacteria. Things we ingest bacteria can continue to metabolize, and things that the bacteria produce we can further metabolize, resulting in a whole array of chemicals that are a result of bacterial-human co-metabolism, whether these are bile acids, indoles, neurotransmitters, sulfur-containing compounds, branched-chain amino acids. We are on it to try to understand the complexity, how bacteria completes our metabolism, adds another dimension to the omics that is so important in defining individuality and response to medications and variation in trajectory of disease that we never imagined before. Resources that have been building up, and this is a fantastic resource, the MIME database, a database for the public to teach us more about the gut bacterial metabolites or gut-influenced metabolites, a partnership between Leiden, the Leiden University with Dr. Thomas Sankemeyer, Nama Karu, who led this with our consortium, contributing to this to create a database for the public. If there are metabolites that we think our gut microbiome produced, how are they produced, where are they found, what do we know about their influences on health, about the exposome influencing these metabolites, as resources for the public, for not only for our initiatives, but also for the public, has been an important core in all of the consortia that we lead. By also embedding people like Dr. Ines Thiele, who does metabolic reconstruction, connecting and modeling bacterial-human co-metabolism, the influences of the diet. These are interacting influences. The diet affects the gut microbiome affects our metabolome, all of this affects our health. So we want to try to understand the interconnectedness of the influences of these omics on our health. Also, big resources that we are building under our gut microbiome initiative and under our Alzheimer's initiative is to be able to map the chemical exposome, these hundreds, maybe thousands of chemicals and beyond in the environment, with 5 centers, again, that are mapping the chemical exposome, giving us an atlas or a roadmap of the pesticides, the metals, all of the things that can affect us from the environment. We are also building with UCSD global databases for foodomics, the food we eat, the components of the food, and the distribution of this and the influence of it on metabolism, big databases for foodomics, big databases on drugs, the drugs we take, the supplements we take, and of course, the microbe mass, the drug mass. And these databases go on. They are super powerful tools to enable us to understand this interconnectedness and inter-influences. Let me give you examples from the Alzheimer's disease. Cannot give it enough justice, but back 10 years ago, President Obama, when he was then president, put a mandate and a series of workshops to change the paradigm in understanding Alzheimer's disease. Billions of dollars are invested. The disease is going to become more expensive than the budget for defense by 2050. Clearly, it's a big burden as we age, and therefore they wanted to change the paradigm from plaques and tangles to a rechange of the paradigm of what Alzheimer's disease is all about by bringing big omics data, putting consortia in a big playground, and enabling and inviting for-profit, non-profit organizations and the advocacy groups all embedded together to change the paradigm in how we understand the disease. Hence was the creation of 6 consortia and 2 enabling large infrastructures to put big multi-omics data from RNA sequence, the genome, the metabolome, the proteome, all of it, to be able to generate big data, make it public in real time, enable us all to interact, to redefine the understanding of the disease. This is over $500 million investment across, and in my own consortium, which is metabolomics centric. We have over 60 individuals from over 30 academic institutions, all contributing to the progress. So in this case, we are tracking, as you see in the bottom, the trajectory of Alzheimer's disease from pre-symptoms all the way across the trajectory. We bring very large studies, such as the ADNI, the large imaging study, ABLE, ROSMAP, Rotterdam. Rotterdam has been a phenomenal resource, and Erasmus partnership is strong, has been embedded in the Framingham Heart Study, the UK Biobank. We want to put all resources possible, map the metabolic changes across the trajectory of the disease, map at every step the influences of the genome on the metabolome, connect the influences of the gut microbiome and the diet, and link all of these metabolic alterations to brain imaging changes, ATM markers of disease, connecting the parts, as the theme of this meeting is truly to be able to say at the end, even the organs interconnecting, peripheral effects central, and it has been remarkable to see how much peripheral metabolism tells us about brain changes, whether, and the changes in imaging and the changes in cognition. It is remarkable how peripheral metabolism taught us about tau pathology, A-beta, tau pathology, glucose metabolic changes in the brain, all the way to neurodegeneration, where we are starting to put a biochemical identity from membrane structure and function, remodeling of membranes, to beta oxidation, to a whole failure of energy metabolism in the brain. What are these links and sensing mechanisms that connect peripheral changes to the brain? We are mapping differences in trajectories between men and women as a first step towards stratifying the complexity and heterogeneity embedded in the disease. Here we are looking at ATM markers of disease, linking the metabolome to these ATM markers, and seeing through the work of our Helmholtz partners how men and women truly are different in their trajectories of disease, in their linkages that connect the parts between peripheral metabolism and the brain. In partnerships with the Baker Institute, we are putting big lipidomics data where we are creating atlases that defines the influences of the genome on the lipidome. We are aligning very large studies such as the ABLE and the ADNI, 1,000 individuals in each and trajectories in each, aligning and seeing if our data replicates. And if these lipid metabolic alterations, which are key central in Alzheimer's disease, can be mapped, replicated, and defined in a better way with influences of the omics. And in this case, we are looking at ceramide, sphingomyelin, oxidative stress, key important paradigms that absolutely replicate across these cohorts, and where we are defining at a granular level the trajectory of the disease, the transition of the disease, and the net influences of all of these factors in transitioning into disease and progressing into the disease. We also map the brain metabolome. What are these metabolites in the brain? Where do they come from? Are these coming from just the influences of the genome? What about the gut microbiome? I can tell you a lot of metabolites produced by gut bacteria are found in the brains, in 500 brains, from the Ross map collection. It blows the mind away. How did they end up there? I'll comment on this as we go along. But we are mapping the influences of the diet. What are the things we ingest? Which of it ends up in the brain? What about the exposome? Chemicals, pesticides, metals, and so on, to redefine our understanding of brain metabolism and the brain metabolome. And here we are giving a snapshot of a beginning of a construction of an atlas for the brain metabolome and changes we see in Alzheimer's patients from bioenergetics, cholesterol metabolism, osmoregulation, inflammation, oxidative stress. And we are connecting the compartments, what we see in the blood with what we see in the brain. Another very important resource in talking about connecting the omics is the first molecular atlas for Alzheimer's disease built by our German partners in Helmholtz as part of our AMP-ED initiative. They are funded, they are partners in this AMP-ED initiative, where they have built a first molecular atlas that is ever evolving on connecting the metabolome, the genome, and the clinical phenotypes in AD. So it becomes a compilation of net interactions between all of these that gives a reader or an experimentalist an ability to go and interrogate this very rich resource into all of these interconnectedness. And it's only a beginning where we are going to add the rest of the omics and the rest of the metabolomics data as we generate it. So it is a resource funded by NIA, will continue to be funded. And of course, it will become global and interactive with a lot. And as we said, not only do we study large cohorts in Alzheimer's, we bring the biobanks and the community studies so that it enables us to go back in time and look at individuals way before the disease happened from the rich collected samples, whether it is the UK Biobank or the Rotterdam study or the Framingham Heart study. All of this enriches the program in a phenomenal way to try to connect information and derive, knowledge. If we want to do translational work, we want to dive deep within a pathway. I want to understand and develop new drugs for Alzheimer's disease. In this case, we have 4 or 5 therapeutic areas that emerged. In this case, to the left, upper left, we saw the importance. Ceramide sphingomyelin is a pathway that has been implicated in AD. We've known this from our work and the work of many others. What we were able to do in here was to bring the power of genetics and metabolomics, lipidomics, to be able to map the intermediates in this pathway, define the influences of the genome, do an MGWAS analysis to define the influence of the genome on these lipid metabolites, add to this flux analysis, add imaging. Imagine this multi-omics approach, plus imaging, plus clinical, all of it embedded together to get a granular insight. Where are these metabolic defects in this pathway? It pointed us to a global disruption with ceramide, with sphingosine 1-phosphate signaling as being key and important. From there, we identified fingolimod, a drug used in MS that targets sphingosine 1-phosphate signaling, and a whole array of compounds that target sphingosine 1-phosphate signaling. We tested those in animal models of Alzheimer's disease. We see the great therapeutic value. We did a metabolic imprint for these drugs in these animal models to learn if they can correct metabolic aberrancies. So you get a detailed map of the effect of the drugs on the metabolome in these animals. And what you see on the upper panel is a full map of the effect of the fingolimod on metabolism, correcting a whole array of things we see as aberrant in Alzheimer's disease, just to give you, including synaptic plasticity, mitochondrial function, inflammation. So just to give you a snapshot how we are moving and translating from the big data, the multi-omics data, into defining drugs and into clinical trials. The gut microbiome is a large initiative. Moving on, is $30 million invested in bringing together great minds with Dr. Sarkis Mazmanian at Caltech, Rob Knight at UCSD, a leader in gut microbiome, and 40 other researchers, including Leiden as well in here, where we are determining the gut-brain axis, trying to understand the complexity of interactions in the gut between immune cells, millions of enteric neurons, immune cells, and trillions of bacteria, and understanding these great number of metabolites produced. This is a snapshot of our Gut Microbiome Initiative, 10 centers in the United States providing samples from Alzheimer's patients. In Project 2, we have large diet, lifestyle intervention, the pointer, a subdivision of the finger large study, in which we show that lifestyle diet intervention are hugely important influences on metabolism and health, and a project where we can model the gut-brain axis and model it mechanistically, and a huge infrastructure that we built over 3 years at the time of the COVID and beyond to put together a whole machinery to mobilize samples, sub-aliquot samples, get them profiled metagenomically, metabolically, genetic, and getting all of this information back in with kits and tools that go back and forth to many centers collecting samples and getting data. Examples of what we see in Alzheimer's disease is an aberrancy in cholesterol metabolism. Not new news. We knew cholesterol is important. What we did not know was the metabolism of clearance of cholesterol by bacteria to produce secondary bile acids. These are the primary bile acids produced by human genes, secondary bile acids produced by bacteria. And what was remarkable is that these bacterially produced secondary bile acids, including the DCA, through enzymatic reactions by gut bacteria, if their level increases, they correlate strongly with brain imaging changes that you see on the right, with degeneration, with cognitive decline. And these bacterial products are all in the brain. When we did sequencing of the brain for presence of bacteria with Dr. Rob Knight, there was no bacteria in the brain, suggesting all of these secondary bacterially produced bile acids in Alzheimer's patients end up in the brain. They are in the brain, probably they are being transported from the blood to the brain. A beginning of a journey. We also determined in anxiety, in studies of depression and anxiety, the second branch of metabolism of production of secondary bile acids, in this case, the LCA from CDCA, this is the second branch of secondary bile acid production. An increase in these bacterially produced bile acids strongly correlated with anxiety, depression, but more so with anxious behavior, right? So the anxiety and all of these behavioral changes could indeed be influenced by the gut microbiome. And we linked all of this to imaging. I don't have time to go through it. Another example is indole metabolism coming from tryptophan metabolism that takes you to serotonin and takes you to canurinine, but also this brown area of metabolism is all gut bacteria that results in production of indoles. Some are harmful, some are good. Indoxyl sulfate is a uremic toxin that is produced from tryptophan by certain bacteria. Yes, that is present. If it is sulfated by the liver, it produces this uremic toxin. If you go and you have bacteria that takes you into another branch, to the indole 3 propionic acid, it's protective. If you increase the levels of this uremic toxin, we have been able to show that it strongly correlates with anxious behavior, strongly correlates with fMRI changes in the brain, in the regions which are affected in depression and in anxiety. And when we manipulate the level of the indoxyl sulfate in animal models with Dr. Sarkis Mazmanian at Caltech and at Stanford with Michael Fishbach, we increase the level of the indoxyl sulfate either chemically or genetically by changing the gut microbiome, implanting overproducers of indoxyl sulfate strains. We see clearly remarkable behavioral changes in these animal models, illustrating the power of this interconnected highways that we are looking at. We are trying to put a map for all of these influences that connect the gut microbiome and the metabolome and their influences on the brain, trying to understand what goes on the brain, what are these processes, a beginning of a roadmap. Another part is the effect of the diet. The diet affects metabolism in profound ways. We have three, as I said, lifestyle and diet. Here I give you one example from a phenomenal team embedded within the U19, where we are studying the effect of a ketogenic diet, 6 weeks on ketogenic diet versus 6 weeks on a low-fat diet for 6 weeks, and then shifting the diets in the individuals. And the most remarkable thing, when we do a deep lipid mapping, with Dr. Peter Meikle and his team, we measure 800 lipids. We look at what happens to lipid metabolism, huge effect of the ketogenic diet, no effect of the low-fat diet. The American Heart Diet does zilch. If I have a problem in the brain, I'm going to be very weary about just taking a low-fat diet. A ketogenic profound effect. Variations among individuals is noted. We do not react to the diet the same way. We are all different in how we change our metabolism. And we revert a lot of the metabolic aberrancies we see in Alzheimer's disease. And we see an influence. We map now the influence of the genome and the gut microbiome. And we see a big influence on the gut microbiome in the paradigm of the effect. We can correct also by using another technology. This is the Nightingale platform, focusing on lipoproteins and some small molecules. We see how we can correct for small molecules, such as the branch-chain amino acids, making the profile much better. But with the ketone bodies that we are trying to fix also, we can increase their level in the blood, not in the brain and not in the CSF, something we are struggling with and trying to sort out. Let's look at depression and the treatment and variation of response. We have the Mood Disorders Precision Medicine Consortium that evolved as a spin-off from the Pharmacometabolomics network that we established to study drugs and effect of drugs. We know we do not react to drugs similarly. We know that a polypharmacy approach is not really acceptable, especially for patients with depression and with hypertension. You give one drug after the other till something happens. Really a very crude, rudimentary ways we need to understand better. And hence we have been a part with NIGMS in establishing the field of quantitative systems pharmacology, where we bring the genome and the metabolome together to tell us in these big studies, 1,000 people, some respond, some do not. We take people at the very ends. The people who are resistant do not respond. The people who respond very well, and then we look at the people in the middle. We do a metabolic mapping. We look at their genome and their metabolome, and we try to learn about how did they respond. I'm illustrating here with the SSRIs, SNRIs, antidepressants, rapid acting. We interrogated the whole class of antidepressants. And the most interesting things, we see response correlating not only with level of serotonin, but with fixing mitochondrial energetics, polyamine, and much more. So the myth that depression is only serotonin is a myth. Depression is far more about energetics and a whole array more that we are just starting to learn. And the most powerful thing we did there, by connecting and doing people at the end, the metabolome, mapping some of the pathways, connecting back to the genome, we were able to determine key pathways that inform the GWAS analysis on who might be a good responder versus a poor responder. We generalize these findings not only from antidepressants, but also to antiplatelet therapies, antihypertensive statins. In every case, we illustrated the power of putting the genome and the metabolome to tell us about variation of response. And Thomas was with us in mapping some of these effects as we do in others. Very quick remarks on depression. I don't have time to really elaborate, but working with the Oxford team, with Cornelia Van Duwen, Najaf Amin, and so on, we are building up on fantastic work that was done, started in the Netherlands, with the BBMRI study in looking at depression, connecting large studies. We are connecting now big data, right? We ended up with the Journal of JAMA Psychiatry, redefining our understanding of depression. Clearly, the food, the diet, and the gut microbiome, big influencers on depression. We don't have time to go through all of this. I'm going to skip just to end up by giving you a couple of new inflammation, where we are knocking boundaries across diseases, bringing together Alzheimer's, Parkinson's, diabetes, autoimmune diseases, putting the whole thing in a queue and asking, is there common mechanisms? Indeed, there are. A lot of common mechanisms. Inflammation, immune dysregulation is one of these common things we are focusing on, again, as 5 consortia embedded to bring the big omics data to understand this complexity. And I exemplify from the oxylipins endocannabinoids pathway of inflammation that highly informs about inflammation, how we profiled some of these oxylipins endocannabinoids, connected the proteome and the metabolome with partners at Emory. illustrated that the key defects we see in Alzheimer's disease using a proteomics platform links and crosstalks to the changes we see with inflammation, replicating all of this work with Dr. Thomas Rankemeyer again to try to connect the dots, understand the transitions, and get to an understanding now of these omics. All of our data goes public in real time. This is a true example of connecting very large initiatives in the United States to try to solve mysteries of the brain. And at the end of the day, it's all about big data, fair data, reusable data. And who is better than the Dutch, who led really the initiatives and walked us through concepts of usability of data. And I end up by saying, there was a vision to build big science. We saw the vision as was a vision for building these monumental things that made people come and visit. And it's no more different, not different with the big data and science. It has been a wonderful journey with a large community over a decade and a half in bringing communities together. And what a wonderful journey and a wonderful family that supported me as I went through in my journey here. And I'm going to stop here.
Thank you very much, Rima. I think we need a coffee break to digest all that information, but let me ask a very simple question, which may be hard to answer. How far are we to translate all the metabolic and lipidomic profiles that you discussed into the clinic that we can use to monitor the progression of Alzheimer's disease?
Very good question. I would say that the transformative step that happened is that the blood, metabolome, and proteome together are highly informative about the brain, which means we can accelerate translation if indeed we can replicate this now globally to say, I don't need a scan, I don't need to go with a CSF or an imaging. I can take a quick blood test. I can map some of these changes, is exactly the translational path for phase 3 of this AMP-AD initiative. I think we have things to translate. Right now, I think starting to test translatability, and I am very confident that tools of picking up things in the blood with proteomics and metabolomics is going to transform our understanding of Alzheimer's and other CNS disorders.
But those are the steps,
so
thanks. Can you put a number there on the number of years that you think this will be, as a proof of concept, proven in the clinic?
Yeah, I think we are putting 5 years, the next 5 years, part of the AMP-AD initiative is to connect the omics. Clearly, the proteome and the metabolome in the blood are key. We also are subtyping and treating. The concept is subtype and treat. So if I see a problem in metabolism, I want to quickly be able to determine in whom is this metabolic defect. A quick test, genetic and metabolic, it could be a SNP and 2-3 metabolites, as we already determined for mitochondrial dysfunction. I want to immediately try to supplement, start with things we can fix metabolically through supplements and changes in enzymatic activities as in inborn errors of metabolism. So I'm hopeful. I think within the next 5 years, we are going to see a lot of translational work, the proteome and the metabolome lead.
Then I propose a deal, Rima. It's now 2024. We will organize the 11th Cross-Omics Festival in 2029. I hope you can be here. And then we showcase basically exactly what you mentioned. Thanks again for joining, and thanks for making the effort at 7 a.m. at your time. Thanks again. Give her a hand, please.
Best wishes.
All right, with that we break, and we break for